Okay, lab 6. The lab 6 will be the last lab you will do before your lab final for this class Biotech 211, and then you will have a week break, fall break, and then you will have the lab, the Biotech 212 started. So it is the last lab, but this lab doesn't have any lab report assignment. Instead, you will be writing actually just a lab notebook, and then you will print out a bunch of results of your primer analysis. At the end, you'll just upload that into the course shell. Okay, so the topic of the lab is designing primers. You know what primers are? Primers are what? Single strand DNA, typically DNA molecules. So single strand DNA that's made out of nucleotides. So they have a unique sequence made up with those nucleotides, and normally those sequences are important because primers are designed to bind to a complementary DNA strand. So when they bind, we call that primer binding process or annealing process. That annealing or primer binding is required for you to be able to PCR amplify the DNA product into a double-stranded form, or you also need that process for your DNA sequencing process. So primers are used in many different places. Okay, so if that's the case, then you need to know how to make a primer. I guess you may not be making the primer, but at least you will be involved in the primer designing process. So your primer will have the correct DNA sequence. Not only that, primer is required to have a certain, the melting temperature. So only your primer would bind to its target site and no other random site. And then your primer should have the certain length to be able to bind on the specific binding region. So it should be around 21 nucleotides around that area. Also, your primer itself should not have a lot of secondary structure. If it has a lot of secondary structure, then it is not going to bind the target sequence. Instead, it may be binding by themselves. So we'll talk about all these things. Okay. So primers are used a lot, so it is important for you to understand the function of the primer and how you design the primer. That's the purpose of this lab. So when you consider designing a primer or a pair of primers, you have to consider many things. Okay, these are the considerations. Okay, number one, annealing sequence or sites. So like I said, the primer sequence should be complementary to its target binding site. Okay, because primer binds to the DNA. When you say it binds on DNA, what does that mean? Its nucleotide sequence is making a complementary base pairing with the opposite target binding site. Okay, so the sequence match should be there. Sequence match means what? A nucleotide on primer will be matched up with T nucleotide on the binding site. G will match up with C. C will match up with G. And then T will match up with A, and that's GC base pairing. Yeah, that is the rule, of course. And then the primer length, like I said, longer the better, because it'll eliminate primer binding on random site. Okay, there's mathematically, very little chance that it's gonna bind on the random site or site with kind of similar sequences. Okay, if your primer is large enough, long enough, maybe about 21 nucleotide, it is very unlikely there will be any DNA sequence that has the matching sequence available within your genome. Okay, so only one binding site will be there. Okay, so the length is important. So it should be large enough, but it should not be too long. If it's too long, there's a chance that your primer will have a lot of secondary structures. We'll talk about what secondary structures are, but if you have a lot of secondary structures, your primer would not bind on the target site. So that's another problem. So the length required size is about 21 nucleotide long. That's probably the requirement. And then TM. TM is a melting temperature, and then from there you can calculate the primer binding temperature condition. Okay, so the TM means what? It's a temperature where your primer would not be able to bind on its target site because the thermal energy is too great. It's going to break up all those hydrogen bonds between your primer bases and the target DNA's bases. The hydrogen bonds are pretty weak, so if there's too much thermal energy, the hydrogen bonds will be all broken up. So TM temperature is enough. So you need to know what that TM temperature is. So when you do the primer binding on its target site, you have to provide the temperature that's lower than the TM, melting temperature, of course, right? Then how much lower? The temperature should be about 5 degrees lower, not too low, not too much lower than TM, because the reason for that, if the annealing temperature, the binding temperature is too low, let's say your melting temperature was 60 degrees, but okay, 60 degrees, your primer will be falling off from the template binding side. Then why don't you use just 30 degrees or 20 degrees to be safe? But you could do that, but there's a problem. The problem is if we use annealing temperature, the binding temperature as kind of too low, there's a tendency that your primer tend to bind at random locations, because your primer is low enough, about 21 nucleotide is made up with that length. There's a tendency that if there are enough DNA sequence match between your primer and the target DNA binding, like 50%, 30%, 20%, 60%, and your primer will still bind on those sites. Okay, we call those random binding sites, right? And then once your primer binds, when you do your PCR will work, right? Between those two primers DNA will replicate. So then you will replicate out so many random PCR products. So that's not going to be good for your PCR purpose, right? So then how do you eliminate those random binding? You eliminate it by providing the primer binding condition at a higher temperature. If you use higher primer binding temperature, let's say the melting temperature was 60 degrees, you decide to use 55 degrees in Celsius as your primer binding, then what's going to happen? So at 60 degrees, your primer would not bind at all. But if you use 55 degrees, yes, it may bind, okay, because it has a unique DNA sequence match, okay. But how about those random binding sites? So the binding that's happening between your primer and the DNA at maybe 50% efficiency, 40% efficiency, 60% efficiency, and those bands will be broken up at 55 degrees because the thermal energy is high enough, it's going to break up all those hydrogen bonds. Does it make sense? So that's why you need to use high primer binding temperature. The reason is to eliminate random binding conditions, and then only the correct binding will take place between your primer and the DNA, the matching binding, like 100% matching binding, okay? So that's why you need to know what the melting temperature is and provide the primer binding temperature at about like 5 degrees lower than the melting temperature. That's what the temperature you're going to use as your primer binding temperature, okay? And I already told you about force primer binding site or random binding site the same way. Okay, any site where your primer binds at less than 100%, those are forced primer binding site. Okay, so your primer is designed to have the matching sequence to its target site, 100% match, right? But if your primer binds with the random site at less than 100% match, maybe 20%, 30%, 40%, 50%, if it binds, then we call those random priming site. Okay, so you can eliminate those by using the higher annealing temperature. Okay, and the secondary structure, what does that mean? So if you have a long primer, okay, like a 21 base pair long, your primer can fold up. Okay, so you can make a hairpin structure. What do I mean by folding up? So your primer is folding up because the bases found within the same primer can pair up between G and C, A and T. So when that happens, you can form a hairpin structure. Okay, hairpin structure. So that's what secondary structure means. Okay, within a single primer, there may be base pairing that can happen. Okay, we call those hairpin structure. Yeah, if your primer is folded up by itself, then that means your primer is not available for its binding with the target site. So that's not good. If we have a lot of hairpin structure within your primer, that's not good. Your primer sequence is not good. And then what's a primer dimer? Maybe your one primer is binding on another primer that has the same sequence, because one primer binds with another primer because there is a matching sequence within them, okay, between them. So that's a primer dimers. So yeah, if you have a lot of primer dimers, what does that mean? Primers are binding by themselves instead of binding on the target site. So that's not good either. So you should not have a lot of primer-dimer possibilities within your primer. So these are kind of the requirements you need to consider when you design your primer. Okay. So designing a pairable primer for your PCR is your task. Okay. So if you know a sequence of your gene or region of PCR amplification, okay, you need to come up with 2 possible DNA sequence sites where your primer should bind. Okay, that is the first consideration. Okay, so the primer will indicate the region of DNA amplification site, because once one primer binds one site and the other primer binds on the opposite side, your PCR will amplify out between those 2 primer binding sites. Okay, that's how your PCR works. Okay, so the sequences of your primers are very important. Okay, then the question is, how do we choose the right amplification site, the primer binding site? Okay, and their sequences. What are the main things that we need to consider when you determine the primer binding site? That's what we're going to study today. Okay, when you do that, yeah, you need to consider all these things we talked about. Okay, not only that, you need to also consider, okay, can I make the PCR products that I need, okay, by designing the 2 primer binding sites. So it should be large enough. It contains the entire gene sequence that I'm looking for. Okay, so when you design your primer sequences, you need to maybe have helpful DNA, the software that can analyze those secondary structures and things like that. So these are the name of the software that you can use, like oligo software, amplifier software, analyzer software, blast search, and things like that. Okay. So let's practice primer designing, okay, using oligo program simulation. That you can get that program from an internet free of charge. You just need to sign up as their customer. There's a company called IDT. They make primers for you, so they have the free oligo analyzer available on their website. But in order for you to use them, you need to just sign up on their website. Okay, you can do that free of charge. And then let's say you have this gene sequence there. Okay, so this gene sequence is from yeast genome. This gene codes for the protein called HSP10. HSP stands for heat shock protein 10. So that's part of my graduate study. I studied a lot of heat shock genes, but anyway, this 10 kilodalton protein encoding gene has that DNA sequence. Okay, so you see the ATG, the start codon, and it ends with TAA, the last codon. Okay, so that's the gene sequence. Okay, so let's say your goal is to PCR amplify out this gene fragment from yeast genome. Okay, you want to do that by using a PCR. Okay, if you want to do that, then you need to design 2 PCR primers that can bind on the beginning region of the gene and the end region of the gene. Okay, so that's the first step. You need to look at the gene sequence, the region of PCR amplification, and come up with the 2 sites where your 2 primer would bind. Hopefully those 2 sites will cover the beginning of your gene sequence and the end of the gene sequence. Okay, so now I highlighted 2 areas, okay, where I'm going to have 2 primers bind to. Okay, so first part is the beginning part of the gene, starting with ATG, from there to here. So it's about 20 nucleotide long. Okay, and the second part is here at the end of the gene, from here to here. I don't know, I didn't count it, about 20 or 21 nucleotide long. Okay, I just highlighted it. Okay, then can I design 2 primers so those 2 primers can bind to these 2 sites? Of course the answer is yes. Then how do I do that? Okay,